Hey biology class, this is Hugh here, and today we are looking at how a neuron works. So this is my not so accurate drawing of a neuron, and there's some terminology you should know already. We've gone over it, but I'll just do it again. So the parts of the neuron: this is the cell body, that's the nucleus. The large part of the cell is here. There are many extensions, the small ones going up this way. They're all called dendrites. Then there's one long extension called an axon, and at the end there's a bunch of little extensions called axon terminals. This is an axon terminal at the end here. This is an axon terminal of the neuron that's before this one. So this is pre. And the signals travel electrically along the cell, but then they have to jump across this gap. And the gap is very important. It's called a synapse. And the jumping, I'll explain how it works, but basically it's not electrical, it's chemical. So there's an electrochemical signaling method where electrical signals travel along the cell. They're turned into chemical signals as they cross the gap. They activate a new electrical signal, and then it goes all the way down and repeats itself. So over here, we would have this would be the dendrite of the next neuron. So with neurons, signaling only goes in one direction. It's always starting at a dendrite, so it jumps the gap, synapse, then the dendrite, down the axon terminal, and then continues. That's the direction of signal flow. Sometimes this terminology is used, so the synapse is so important we talk a lot about it. So this would be called, this dendrite is also known as the postsynaptic membrane. And that would make the axon terminal the presynaptic membrane. So to understand how the neuron works, we have to understand how does the signal travel along the cell, then how is it converted into a chemical signal, and then how does that activate a new signal in another neuron. So let's take a look in a bit more detail. Okay, so next we have to look at what are called the transmembrane proteins. These are all proteins that are embedded in the cell membrane of a neuron, and they're also found in many other cells, by the way. And this is a great example of reviewing some of the things that you studied when we did the mitochondria. Remember that there are pumps which do what it's called active transport. To be a pump, it means it needs energy. And always a form of energy is called ATP, adenosine triphosphate. And this is called the sodium potassium exchange pump. So it pumps ions across the concentration gradient or up the concentration gradient. So it needs energy to run. And remember that sodium, if you haven't done chemistry, sodium is symbolized as Na plus, and potassium is K plus. So they're both positively charged ions. And whenever this pump runs, it pushes outside. So this is out of the cell, and this is in the cell. It pushes sodiums out, and for every time it runs, it takes 3 sodiums and pushes them out. And at the same time, it's an exchange pump, so it brings in 2 potassiums. So as it's running, using energy, it's going to put more sodium outside the cell and more potassium inside the cell. And also it's going to increase the amount of positive charge outside the cell, because whenever it's running, it's bringing 3 sodiums out for only 2 potassiums in. So this is a pump. This is active transport. The next, the remaining elements here are all channels. They participate in what's known as facilitated diffusion. And so here's the K plus channel, and just to give it some context, I'll put the cell membrane in its place here. There's a phospholipid bilayer. So these guys sit inside the phospholipid bilayer membrane, and these guys have room to let a potassium go through. Remember one very important thing, no positive ion or negative ion can cross the membrane without assistance. This part, remember, is hydrophobic. Where this is hydrophilic. Philic. It's hydrophilic out here, it's hydrophilic inside, hydrophobic in the middle. And because ions have a full charge, not even hydrogen ion, not even the smallest one can get through. So the only way potassium's getting in or out of the cell is through one of these guys, except for, of course, here. So it's got to have assistance. The channels, these are channels. And they let specific ions through. Okay. This process that when you need an assister, a facilitator, to get something in or out of the cell, this is facilitated diffusion. So it's an example of what's called facilitated diffusion. And for this one, wherever the concentration gradient is greatest, if there's more potassiums inside, they leak out. If there's more potassiums outside, they leak in. So it just goes whatever direction. So there's no energy involved. It is not a pump. It simply lets things flow. The next three are all examples of the same kind of thing. They're all channels, but they're known as what are known. So gated channels can be blocked. In their default setting, they have a little blocker which stops anything from getting through. The two green ones are sodium ion channels, and the blue one down here is a calcium ion channel. So this would be a gated sodium ion channel. And so is this. Where this is a gated calcium ion channel. So the two on the right are gated in the same way. There's a little lightning bolt, which means these are called voltage gated. And there's this one. And the key to opening the gate is in terms of a change in voltage. When there's a change in voltage in the proximity of either of these proteins, that pops the gate open and lets the ions through. Remember, when it's open, in the green one, only sodium, only Na plus can get through. If there's potassiums or calciums out here, they are not allowed through. No way. Only sodium's allowed through, and it's only allowed through when the gate's open. And in the same way down here, it's only calcium 2 plus, only that can go through when the gate's open. So those are both voltage gated channels, and again, changes in voltage in the membrane surrounding the protein cause them to pop open. When the voltage returns, they'll just close. Over here, this is a different kind of thing. It's got a cup here. This is called a ligand gated channel. And a ligand is, in chemistry, a term that's used for a small molecule and the ligand fits in to the cup. So if the ligand's like this, L, like that, it fits in. And when it does so, it pops it open. And probably you're now thinking, well, that sounds a lot like how enzymes work, and it is. It's very specific. So just like the active site or the allosteric site in an enzyme, this shape of this molecule fits this particular cup. There are different ligands for different types of ligand-gated channels. These ligands in this context are known as neurotransmitters. So the neurotransmitter is the ligand, and it is the thing that activates this gate. When it's present in the cup, pop the gates open, and then you're out in business. Neurotransmitters are quite well known, and there's a class of them. There's a number of different types. So it's not just one molecule, it's a group of molecules. But say, for example, if this was GABA, which is one of the neurotransmitters, it would click in and pop it open if this was a GABA, a ligand-gated channel. But another one called serotonin would not work, but it works on the serotonin type one. So again, shape specific, just like in enzymes, super important. Okay, so now I've attached these guys to my picture of the neuron. Now, this is a big one. That's the axon terminal of the neuron. So this would be the previous neuron. Signal will travel this way. Presynaptic membrane, postsynaptic membrane, and this, of course, is the synapse. I'm not going to worry about how that all works right now. What I want to do is focus on the neuron and to explain how it gets ready to take a signal. It can't always take a signal, so it has to be prepared. And what it does is because it's running these pumps, the sodium-potassium exchange pumps, they use ATP, they're always on. And so they're always pumping out the 3 sodiums out of the cell. And at the same time, they pump into the cell the potassium ions. Okay? So as this pump runs, and each one's the same, they're always pushing sodium whenever it's found inside the cell out of the cell. So you're getting a lot of sodium ion out here, lots and lots of it. And because the pumps run all the time, 24/7, that's why your brain needs so much sugar all the time. They're always pushing sodiums out. They're always bringing potassiums in. And it's not quite an equal exchange, so that gives you more positives outside. But what's even more impressive is that, remember those leaky channels, the facilitated diffusion channel for only potassium ions, once potassium's pumped in here, all the potassiums, they have a higher concentration inside the cell than outside the cell. There's less, so they will simply diffuse out of the cell. So even more positive ion comes out of the cell. So the cell, the potassiums diffuse outwards, and then they're, of course, picked up and pumped back in. But the result of everything is you're always losing positive charge inside the cell, and you're gaining positive charge outside the cell. So the cell actually will have, now it's not just these sodium pluses, but it'll have a positive charge on the outside of the cell. And because you've removed positives from a neutral environment, it's got a negative charge inside the cell. When that's set up with negatives on the inside, positives on the outside, this is called the resting potential. So when it's set up so that it has a lot of positive charges everywhere outside the cell and negative charges inside the cell, then it's ready to take a signal. And if the simple way of knowing what is the negative charge is that all solutions are neutral. So let's just say it's a salt solution. Salt is NaCl. It's sodium plus chlorine 1 minus chloride. So for every Na plus, there's a Cl minus. So we could imagine all those negatives are chloride ions. So the idea is, as the cell is working, it's always pushing positives out in the form of sodiums. It's bringing in positive potassiums, but then letting them leak out. So the entire outside of the cell has a positive charge, and the entire inside of the cell has a negative charge. And in the next video, I'll explain what use that is.